Good afternoon, and thank you for standing by. All lines are on a listen-only mode until the question and answer segment of today's conference call. At that time, you may press star followed by the number 1 to ask a question. We will be taking questions via the phone only on today's conference. Today's conference is being recorded. If you have any objections, you may disconnect at this time. I would now like to turn today's conference over to Ms. Irene Ihier. Thank you. You may begin.
Thank you. Hello, and welcome to today's FDA webinar. I am Irene Ihier of CDRH's Office of Communication and Education. On July 6, 2016, the U.S. Food and Drug Administration issued 2 draft guidances that, when finalized, will provide a flexible and streamlined approach to FDA's oversight of next-generation sequencing tests. These 2 draft guidances support President Obama's Precision Medicine Initiative, PMI, which aims to take advantage of the progress made in genomic testing to accelerate the development of new treatments that take into account individual differences in people's genes, environment, and lifestyles. The purpose of this webinar is to share information and answer questions about the technical and regulatory aspects of the guidances. Your presenters are David Litwack and Dr. Laura Kuntz from the Personalized Medicine staff here in CDRH. Following the presentation, we will open the line for your questions related to technical and regulatory aspects of the guidances. Additionally, other center subject matter experts will be joining the Q&A portion of our webinar. Now, I give you David.
All right, thank you, Irene, and thank you to everybody who called in for joining us today to discuss the 2 guidances on next-generation sequencing that FDA published on July 6th of this year. We will use these webinars to provide more detail on these guidances, and particularly here to focus on the technical and regulatory aspects of the guidances and use this opportunity to address questions you may have and clear up things that you may not have understood in the guidances. So here in this hour, we will provide background that motivated the background of describing the motivation for release of these guidances, and then to dive into the details of both guidances. And then we will discuss what this means for the regulation of next-generation sequencing and how we will proceed after these 2 guidances are finalized sometime in the future. And we will provide time for questions at the end of the webinar. I will discuss the first guidance on the analytical standards for next-generation sequencing, and then Dr. Kuntz will take over and address the guidance on the use of databases. So first, to some background. Why did we release these guidances? Why did we write them? So as many of you may be aware, President Obama announced the Precision Medicine Initiative in January of 2015. This effort is designed to enable a new era of medicine through research, technology, and policies that empower patients and providers to work together and researchers toward the development of individualized care. Well, the centerpiece of the PMI is really the million-person cohort, the PMI cohort program, which is going to engage 1 million participants in research that will advance precision medicine. The PMI also recognizes that the success of research in precision medicine and its translation to the clinic relies on diagnostic tests that are both safe and innovative. And probably no diagnostic test has been more central to the advance of precision medicine than tests that use next-generation sequencing, or NGS. So we've recognized that NGS has unique features that makes it in many ways different than other diagnostic tests. For this reason, as part of the PMI, FDA proposed to optimize its regulatory oversight of NGS tests by developing an approach that takes these unique features into account. The goal of doing this is to help assure that NGS tests provide accurate and clinically meaningful results and to promote innovation in genomic testing. We believe these 2 goals are intimately linked and that successful innovation depends on accurate and reliable tests. and that appropriately designed oversight in the space can achieve both goals simultaneously. We have previously discussed the reasons for our proposals and the concept underlying the proposed approach in a series of white papers and workshops, and you can find these discussions on FDA's website. In this webinar, I will give you just a brief overview of the goal of the optimized approach before diving into the details of the two guidances. So conventional diagnostic tests are typically based on science or technology that has come to some sort of answer that is in some sense settled, an improvement, and also technology that has been investigated and locked down before coming into the clinic in many senses. And although there can still be improvements and advances in those technologies and in that science, they tend to happen more discreetly with varying intervals of time, marked by stasis. FDA's system of pre-market review is well suited to this and has a long track record of success spanning over 40 years. This system has also accommodated those individual NGS tests that have already been reviewed by FDA. As a class, however, NGS is still undergoing rapid technological evolution, and the evidence supporting the clinical use of these tests is also rapidly changing. This essentially reflects the state of the field of precision medicine and the fact that it is undergoing as a whole is undergoing rapid development. So for this reason, FDA believes that pre-market submission and review for each test modification and each change in evidence that would necessitate maybe a change in the variants a test was purported to detect would not keep pace with innovation. Instead, FDA proposed a system that would be, as part of the PMI, that would be more responsive to rapid changes in the field of precision medicine. The draft guidances published this month represent a critical first step in the development of such a system. Any different approach, though, must guarantee the same assurances that are provided by FDA pre-market review. An FDA review of any IVD typically focuses on three elements. The first is analytical validity. Does the test accurately and reproducibly detect the intended analyte? And for an NGS test, does it detect a variant when one is present and not detect a variant when it is not present? And this is typically demonstrated by bench testing with the data typically provided to FDA by a test developer or sponsor. The second is clinical validity, i.e., when you detect the intended analyte or analytes, are you correctly identifying the disease, condition, or state of health? And in NGS, this is often related to the clinical interpretation or classification of the variant. For IVDs, demonstrating this does not usually require randomized clinical trials, but instead clinical validity is typically demonstrated by literature, retrospective studies, or other sources of evidence. Finally, FDA is looking at labeling or what claims are made about the test, because labeling has to be truthful and not misleading. It should reflect the analytical and clinical evidence supporting the performance of the test. Now, all of these elements are reviewed in light of the intended use of the test. So, for instance, an NGS test intended to detect circulating free DNA may require a higher sensitivity than an NGS test intended to detect germline variants for the diagnosis of hereditary disorders, and thus the bar for analytical validity might be separate in those two cases, even though the core technology might be very similar. All right, so given that background, now let's move to the first of the two guidances, which is the first of the two guidances, which focuses on the analytical standards and how they might be used in the review and oversight of NGS tests. I should say before I go that taken together, these two draft guidances are designed to anticipate and support the needs of rapidly evolving NGS technologies. We want FDA regulation or oversight that can support reliable, accurate, and understandable test results while promoting an efficient path to market for all test developers. So again, the first guidance describes how standards can assure test quality and describes a potential regulatory path for certain uses of NGS. And the second guidance discusses how FDA and test developers could leverage third-party genetic databases to support the clinical validity of genetic variants. Both of these guidances rest on the foundation of transparency, that NGS test performance and the evidence supporting the clinical interpretation of genetic variants should be available to users of these tests. It's important to point out that these draft guidances are proposals released for public comment. They have not been finalized, and they do not imply any current requirement that anybody must meet. Nothing has changed at this point in time. I also want to note that both proposals, if finalized, describe voluntary paths. We intend for these to be alternatives to pre-market review, and it will be up to test developers to select the option, pre-market review or a so-called PMI path that best meets their needs. Finally, because these are draft guidances, we critically depend on your public comment, and there will be many opportunities. We've opened a docket, and we'll have links to those dockets at the end of this presentation. Okay, now on to the draft guidance, which is the use of, first one, the use of standards and FDA regulatory oversight of NGS-based IVDs used for diagnosing germline diseases. One of the first issues we had to grapple with, I would like to point out that, just even before I get to the scope of the guidance, that these guidance as well as the database guidance were drafted after extensive interaction with public, with experts, with people who use these tests, who develop them and who run them. When we observed during that process much variation in how tests were used. And one of our goals here was to accommodate that variation. One of the first issues we therefore had to grapple with is how to think about different uses of NGS, because it is used for such a wide range of purposes: diagnosis, risk prediction, health and lifestyle, targeting treatment, and so on. And when used for diagnosis, even in like for a single use, a broad use like diagnosis, there are still a wide range of tests. Those for diagnosing specific diseases like cystic fibrosis, those that may diagnose conditions like hearing loss to identify which variant may be causing that, and those that are unbiased searches for the causes of disease related to a constellation of symptoms. For our purposes, it made sense to ask which uses might be covered by a single standard, and we ended up binning tests into broad intended uses, and this slide depicts some of those bins. This first draft guidance focuses only on one bin, the one titled hereditary disorders. We have done this because of the complexity of trying to address all uses at once. Also because we believe that most of the indications within this bin are moderate risk, and that it is, in many ways, the technically simplest case. We believe that this is the appropriate starting point. Hence, this guidance applies specifically, and as stated in the guidance, only to targeted or whole exome sequencing NGS-based tests intended to aid in the diagnosis of individuals with suspected germline diseases or other germline conditions. Throughout the rest of this webinar, in this section, referring to this guidance, where I'm referring to an NGS test, whether or not I say it, I will be meaning to refer to NGS tests that fall within this broad use. Note, however, that in the future we will be expanding this approach to other intended uses, and it is a high priority for us to do that to cover oncology, liquid biopsy, disease risk, and all of these other uses. Okay, so the technical recommendations that we provide in this guidance describe an approach to test design. This is done to accommodate different test designs, components, indications, and so on. Again, given the wide degree of variability in running NGS tests, it was important for us to preserve that feature as we believe it's important for innovation. The technical recommendations that we provide can form the basis for future FDA-recognized standards or special controls, and I'll get into that a little later. So the overall approach that we propose is, they aren't standards at this point, but essentially what we're calling design standards or design recommendations or considerations. The idea here is, again, this is a process of test development. It is not meant to specify specific components or largely specific performance characteristics, although there are a few exceptions to that. The idea is to first, in designing the test, identify the intended use and your user needs, to use that to set performance, identify performance metrics that the test must meet in order that are required for that intended use, and to identify performance thresholds that that test must meet for each of those metrics. Once you've identified the necessary performance, and this also includes the sort of test run quality metrics we see in NGS that identify whether an individual run may or may not be accepted. This could include things like coverage. We'll get into that again a little bit later. You then actually assemble the test, put the components together, write the SOPs, and then you validate to determine whether that test actually meets the predefined performance thresholds. If it doesn't, you have to modify, so you go back to the beginning with test design and repeat until you have designed a test that actually meets the required performance and hence can perform according to its intended use. Okay, so test design. The intended use of, the first thing you do is identify your intended use. Are you going to be testing for undiagnosed diseases in general? Will it be for hearing loss? Will it be for cystic fibrosis? We also separate out user needs because sometimes there are needs that will affect test design that wouldn't be part of a traditional, intended use. For instance, you may be receiving samples that may undergo extreme conditions under shipping, and your test must be able to accommodate those. Or you may have a certain turnaround time. It may not be in part of your intended use, but that still is going to factor into test design. Specimens, you must identify what specimens you expect to be testing. And also for genomics, for NGS tests, what are the interrogated regions? What part of the genome are you going to be sequencing? Will it be specific genes, specific parts of genes, whole exomes? That all must be defined. From there, you set, again, your performance needs. What sort of accuracy might you need? What sort of precision? And then you move on to identifying the components and methods you need to design a test that meets that performance. Now, all this feeds back. Sometimes, for instance, you may be limited in your available components, and that may affect the types of uses you can have. You may have, it may be that certain sequencers have certain biases that prevent you from reliably detecting certain variants. And so that may, if test components and methods are a limitation, that may affect how you, the use of your test. So if we drew all the arrows, this would be a confusing diagram, but this is an example of how, in many cases, one of these bins may influence what happens in another bin, and it's very much a cyclical process. So So the test performance, let's get a little more detail into, let's provide a little more detail about that. So what we're talking about here is, again, identifying performance metrics and setting minimally acceptable thresholds. The guidance specifies 4 overall performance metrics that we believe should always be assessed. Accuracy, precision, so this is reproducibility and repeatability, limit of detection, and analytical specificity. And I want to call out here, this analytical specificity includes cross-reactivity, which we know is an issue with pseudogenes. It's the reason why some regions are difficult to sequence, as well as cross-contamination between patient samples and the ability to detect that. In some cases, we recommend minimum performance thresholds, but not in most. We believe that a minimum accuracy limits could be set higher, but they should be at least 99.9%, and that a confidence interval around that should be reported, and that a precision no less than 95% should be obtained. In many other cases, though, we aren't going to set a value, and that's one that a test developer would have to select and document the justification for that. Test run quality metrics. So as I said, these are metrics that assess whether a test run or variant call should be accepted on a run-by-run basis. And we recognize that different test developers use different metrics, or even the same metrics for different purposes. And so we only specify coverage as the one metric that developers should always use in some fashion, but then there are other metrics that we discussed that can be selected by test developers as the need arises for specimen quality, DNA quality, and processing, sequence generation and base calling, and other types of metrics. But those should be selected and justified, but there should be enough. The metrics that should be used should provide assurances that that a test run can be accepted, or confidence that a variant call is correct. General recommendations for performance evaluation studies. Once you've identified your performance metrics and the thresholds, you then have to, and you put your test together, you have to do validation experiments. And we define many elements that should always be included in validation experiments. Some of the ones I want to stress here are evaluating end-to-end performance, and that's just not a part of the test. Validating across representative genomic regions, variant types, et cetera, that are relevant to what you are, your indication. Identifying the types of sequence variants, the limitations of the test. What can you not detect with adequate accuracy and precision, and using appropriate specimen types in your validation studies, and where you use plasmids or synthetic constructs, performing commutability studies to assure those are going to give you adequate validation results. We don't specify the number of specimens required for validation experiments because that's going to vary depending on the situation, but test developers have to be able to determine, confidently determine the number of specimens required for validation experiments. So accuracy studies, I just want to make a point that it's important to have a comparator method. Sometimes it's Sanger sequencing, sometimes it might be something else, and in some cases you can also do it in the absence of a comparator method. You can also use a comparison to test-generated, compare a test-generated sequence to a consensus sequence of agreed-upon, well-characterized samples. And genome in a bottle, NA12878 would be an example. We expect that all results would be, validation studies would be documented with confidence intervals, not just point estimates, and broken down by variant, sequence context, and other variables. Other recommendations are listed in the guidance. We have other recommendations, and I won't go into detail. The use of supplemental procedures like Sanger orthogonal confirmation, which may affect how validation is conducted, but they aren't really considered part of the core process of the test. And we discuss those, but it is up to the test developer's discretion whether that would be sort of part of the test process or not. Variant annotation and filtering, presentation of test performance and test reports, and modifications as well. We do want these standards to encompass modifications. Regulatory considerations. So in addition to the technical recommendations that we put forth, we also describe potential regulatory paths. And we describe the regulatory path that we think will get us to begin to achieve the overall PMI vision that we've laid out, which would be a standards-based approach for assuring analytical validity. So what currently, we have a system of classification for IVDs, with Class 3 being the highest risk tests that require pre-market approval. Currently, NGS-based tests are Class 3 by default because we have never cleared or approved NGS-based tests for the intended use we are discussing here. However, we believe currently it may be possible to classify these tests as Class 2 devices. And we outline what FDA believes is needed to support this classification. And we discuss the possibility to go a step further, that in the future we could even exempt these tests from pre-market review. And this is a process we have used in the past for other IVDs. So the general need here to be able to do this, to get to Class 2, is to obtain a de novo submission. And if the de novo pathway is one that allows, it's a type of submission to FDA that allows FDA to review and down classify, if appropriate, a new type of technology, a new type of test. In this case, we believe, and state in the guidance, that it's likely that if we received a de novo for this intended use, we could come to a Class 2 classification decision, in which case the technical recommendations that we present could act as special controls. Special controls are just special requirements that a test would have to meet, and so those special controls, we could do it that way. Now, we could also come to a decision that future tests would have to comply with the special controls, but not even have to come into FDA with a pre-market notification. We would need evidence, justification for that, but that's something that could be done in the context of a de novo at some point. And that would allow us to implement standards, whether in special controls or through FDA-recognized standards that could be based on these technical recommendations without without pre-market submission. So with that, I will leave it and hand it over to Dr. Kuntz to discuss the database guidance.
Thanks, David. I realize we're hearing that some people are having trouble accessing the slides, and we're working on figuring out what that issue is. But the slides will be available along with a recording next week. So apologies for that. Anyway, my name is Laura Kuntz, and I'm a member of the personalized medicine staff in the Office of In Vitro Diagnostics here at FDA. And I'll now spend a few minutes discussing our second draft guidance document, the use of public genetic variant databases to support clinical validity for next-generation sequencing-based in vitro diagnostics. The guidance outlines the agency's thinking about how genetic databases can be used in a regulatory review of NGS-based tests, and that is how public genetic databases that follow certain quality specifications can be sources of valid scientific evidence to support the clinical validity of NGS-based tests. So how does the ability to tap into publicly available genetic databases to support clinical claims benefit patients? Databases of genetic variants have the potential to speed evidence development for NGS-based tests, since the evidence housed in these databases is typically generated by multiple parties. Collectively, we can obtain evidence for the clinical interpretation of a greater portion of the genome than we can individually. Crowdsourcing evidence generation in this way eases the burden on any single test developer and could speed new tests to market. Importantly, aggregated data could also provide a stronger evidence base for NGS-based tests. Tests that use these databases would be connected with the current state of scientific knowledge regarding a genetic variant and its relationship to a disease or condition. Finally, FDA believes that as more evidence is gathered and improved, new assertions supported by the evidence within a database could be made. Next, I'd like to take a minute to outline the guidance before we dive into some of the specifics. To allow NGS-based test developers to leverage these databases, FDA has published its draft guidance which lays out how genetic variant databases can be used as sources of valid scientific evidence to support the regulatory review of NGS-based tests. Specifically, the database guidance lays out a series of recommendations that FDA believes, when followed, would allow the database to be considered a source of valid scientific evidence, which can then support the clinical validity of the test. Database administrators that run databases that would meet these recommendations could voluntarily apply for FDA recognition. And I just want to stress, again, voluntary. They would then submit an application to the FDA demonstrating that their database meets these recommendations set forth in the guidance. We will discuss these recommendations in greater detail in a minute, but broadly they rest upon three pillars. One, transparency. Two, validated processes and formal procedures. And three, expert interpretation. Lastly, I want to note that because transparency is a key component of these recommendations, the guidance document only applies to publicly accessible databases, so not proprietary databases. I'd now like to discuss our quality recommendations and the FDA recognition process. Because databases must demonstrate that quality recommendations have been met in their application, I will just discuss the recommendations in the context of an application for FDA recognition of a database. To become recognized by the FDA, a database administrator would have to demonstrate that the recommendations had been met. This recognition would occur in three steps. First, a database would submit a voluntary application to the FDA for recognition, and that is at the discretion of the database administrator. FDA is not compelling any database to seek recognition, and this process is purely voluntary. Second, the FDA would assess the genetic variant database policies and procedures for obtaining and maintaining data and making variant assertions. This would include evaluating the database to ensure that it provides transparency regarding its data sources and operations, including versioning and the use of publicly accessible SOPs or standard operating procedures, that it provides sufficient assurances regarding the quality of the source data, including the use of accepted nomenclature and clear documentation of the evidence used to make variant assertions, that it has SOPs that define how variance information is aggregated, curated, and interpreted, and further, that the SOPs that outline how assertions about a variant are made are based on validated decision matrices. That data is collected, stored, and reported in compliance with all applicable regulatory requirements regarding protected health information, patient privacy, research subject protections, and data security, and that variant assertions are made by qualified experts, and these experts adhere to the database's conflict of interest policies and that they disclose any conflicts. It's worth pointing out that this is an incomplete list of recommendations, and there are additional details and recommendations in the guidance. And then finally, as part of the recognition process, FDA may spot check variant assertions to assure that they are made in accordance with the database's SOPs and reflect the current state of scientific knowledge. Now, once the database has been recognized, the third step is maintenance of that database's recognition. This maintenance of recognition would occur through periodic reassessment of the database, its SOPs, and its assertions. So once the database has been recognized, FDA believes that the evidence contained in that database would generally constitute valid scientific evidence that could be used to support the clinical validity of next-generation sequencing-based tests. The assertions that a database could make about a genetic variant could include a variety of different variant types, such as relating to the pathogenicity of a variant or whether that variant may be related to a certain response to a certain therapy. At our public workshops, we heard that it was important for patients and providers to receive information regarding variants of uncertain significance, as these may be medically useful information for an individual patient. Therefore, we have proposed that it would be permissible to report this type of information, but we'd like to receive public input on that point. Regardless of the type, all variant assertions should be supported by adequate information detailing the evidence within a genetic database supporting that assertion. For example, how many times has a variant been seen in people with a certain disease or condition? What scientific data links a gene to a disease, and what is the biochemical nature of a variant's effect on encoded proteins, and so on. Those assertions should also be an accurate reflection of the current state of scientific knowledge and generally would integrate multiple lines of evidence. And finally, all assertions should not be false or misleading. We believe that these recommendations dovetail with those regarding test reports and the analytical standards guidance that David mentioned. Those recommendations state that, among other things, the relationship between a reported variant and the clinical disease or condition being assessed must be clearly stated. In summary, this guidance document outlines an approach that allows test developers to take advantage of the benefits of data aggregated in FDA-recognized databases to support the regulatory review of their test, and it outlines a pathway for databases to voluntarily seek recognition. Next, I'd like to summarize the two guidances and discuss their future implications. Taken together, these guidances discuss the possible future down classification and exemption from pre-market review of NGS-based tests that demonstrate conformity with the standards outlined in the analytical guidance and use assertions from FDA-recognized databases. FDA believes that this potential approach offers speed, scalability, and safety. So speed. This approach, when finalized, would provide test developers with an efficient path to market and connect patients with tests quickly. Scalability. Test developers, both large and small, can benefit from this approach because it is based on standards and evidence that everyone can access. And safety, that this approach encourages innovation while still assuring patients and healthcare providers that NGS-based tests are safe and effective and that they provide accurate and meaningful results. So next steps. Currently, the draft guidance documents are open for public comments until October 6th. During the public comment period, we also plan to hold a workshop to discuss the guidances, and that will occur later this fall, and we hope to be able to announce that date soon. So keep your eyes peeled for that. Following closure of the open comment period, we will, of course, analyze comments, make any necessary changes, and hopefully move towards finalization and implementation. Eventually, we also hope to expand this approach to other intended uses of NGS-based tests, such as for oncology, and we'll have opportunities for the community to comment on additional standards or criteria that would be necessary to assure the safety and effectiveness of these other types of NGS-based tests. And so, as I mentioned, the comment period for each of these guidances is open now through October 6th. We hope that you will submit comments regarding the substance of each of these guidances, what we got right, what we didn't, what you think we should change. Each of these guidances is also accompanied by a Federal Register notice that includes specific areas that the FDA is seeking public comments on, in addition to whatever else you'd like to comment on. And so as you are developing your comments, I encourage you to look at these questions and offer your feedback on them. And then finally, before we get to the Q&A portion, I'd like to put in a plug for additional ways that you can be part of the process. To fully realize the vision outlined in these draft guidances and expand it to other indications in the future, we need your help. We need community input on the technical recommendations of potential standards for other uses, like oncology, and for standard developing organizations to issue consensus standards for NGS tests. Following our involvement in the creation of the Genome in a Bottle reference genome, FDA continues to work on the creation of reference materials in partnership with other public and private organizations. FDA encourages community aggregation of genetic variant information and supports data sharing efforts necessary for the creation and continuation of sustainable, high-quality databases. And finally, curation. As we've heard at all of our public workshops, databases are dependent upon external experts to assist in curation and interpretation activities, and there's always more work to do. So please consider getting involved there too. And now I'm happy to turn it back over to Irene.
Thank you. We'll now take questions.
Thank you. At this time, if you do have any questions or comments, you may press star followed by the number 1. Again, that is star 1 to ask a question and star 2 to withdraw your question. Carol Ryerson, you may ask your question.
I was looking for information on accessing the slides since they weren't available to us during the presentation. So I think that was addressed, that we'll be able to access them on the website in a week.
No, hi, my apologies. You can actually access them now on CDRH Learn. We have a slide, well, I'll go ahead and read the address for you. We apologize again. That is www.fda.gov/training/CDRH Learn. And when you get to that page, you can actually find the slides under the heading Specialty Technical Topics and the subsection Device-Specific Topics. Again, we do apologize for participants not being able to access the slides via my meetings.
We do have a few more questions. Gloria Lee, you may ask your question.
Hi, this is a question for Dr. Litwack. It's more of a clarification question to a statement that you had made. You had said that when final, the analytical standards NGS guidance is intended to be an alternative to pre-market review for NGS test developers, and they can decide to follow a traditional pre-market review or the PMI path. So my question is, does that mean if the test developer follows the design standards listed in the draft guidance once it becomes final, does that no longer include the QSR requirements for design controls, et cetera?
No, we expect that the general controls, including QSR, would still apply in any situation. These are meant to sort of be the equivalent of special controls, you know, to guarantee analytical validity.
Okay, thank you.
Thank you. Our next question comes from Ash Carr. You may go ahead.
Hi. Thank you for taking my question and presenting this emerging topic. I would like to ask the panel where can a test developer locate a list of FDA-recognized databases? Thank you.
I'm sorry, I think you broke up a little bit. Could you try to repeat the question?
Sure. My question is, where can a test developer locate a list of FDA-recognized databases?
I think the question was, where can you access a list of FDA-recognized databases? At this time, there are no FDA-recognized databases. With the draft guidance, we hope to move towards finalization and have databases come in to be recognized. That process, again, is voluntary. So it would be at the behest of a database wishing to come in and be recognized. But no, at this time, there are no recognized databases. At the time that we do recognize them, we'll put them on our website and make sure that that information is available to everybody.
Okay, thank you.
Mm-hmm. Thank you. Laura Bean, you may ask your question.
Hi, I have two questions. The first is, do you plan to provide laboratories with a template for submitting this information? And then secondly, in regards to databases, once you have an approved, recognized database, how are you viewing the assertions within that database? Are you viewing this as information that may be helpful to a laboratory, or are you viewing these assertions, which may change over time as more information becomes available, are you viewing this as the appropriate assertion for a variant?
Hi, this is Liz Mansfield. I'm the Deputy Office Director for Personalized Medicine here. And at this time, we don't anticipate supplying a template because it's not clear to us that a template would be appropriate for this, but people may address these standards in different ways. There's usually an opportunity to address standards in different ways, and there's an opportunity to deviate from the standards with appropriate justification. So generally, we don't use templates. Although if you would like us to, I would certainly encourage you to include that in comments and perhaps provide some more detail if you can. The second question.
In this case, if people are submitting their groups of genes or it's variations on the same test with the same basic data with different gene lists, and it may be appropriate in this case.
Okay. And the second question was about how we view the assertions in the database. We expect that databases, when they are used to support clinical validity claims, will be versioned so that we will know at what point you reference that database and can refer to the assertion at that date. We do expect that people would be able to update their assertions based on changes in the databases. In general, we hope that we can find a way for that to be done without returning to FDA for any kind of clearance or approval, so that it would just be it would be an ability to update sort of in real time.
And would you like to go to the next question?
Yes, please.
Karen Thacker, you may ask your question.
Oh, sorry. I think you're referring to Karen Thacker from Greystone Oncology?
Yes,
Hello?
your line is open.
I'm sorry. This is a colleague of ours. So I'd like to know when you expect to have a draft guidance for tumor sequencing.
We can't. I couldn't project a specific date, but what we would like to do is see the public comment that we receive on this, because we think this would form the core of any recommendations for tumor sequencing or other oncology-related applications. And then I think definitely it's one of our priorities to then extend this to semantics. So we hope it'll be one of the next things we can do. But, you know, we want to wait and see what the comments are on this sort of simpler case. We recognize that for somatic mutations, there are special technical considerations that we have to build into this.
Great. Thank you.
Thank you. Lori Lee, you may ask your question.
Hi. I have a question on the previous presentation. I think it's great that this guidance document is getting out. Regarding the 4 metrics, that is accuracy, precision, LOD, cross-contamination, and cross-activity, are more specific guidelines expected to come out in view of different aberration types and so on?
So we had a little difficulty hearing you, but I think your question was, do we expect to put out additional guidances based on, with more specific information based on different types of genomic variants?
Yeah, specifically for the 4 metrics that were laid out in the previous presentation, precision, accuracy, LOD, and cross-contamination filters.
What we're really hoping will happen is that the community will develop standards that address these various areas. If it's something that's critical to your test and the way that you're running your test and the intended use of your test, then we would in general expect that you would address it in some way. I don't believe that we plan on putting out additional guidances at this time.
But let me also follow up and say, if there's specific information you believe we ought to provide, we would like to receive that in public comment.
Got it. Thank you.
Thank you.
Thank you. Elise Cuban, you may ask your question.
Hi. So my question is for Dr. Litwack. So I think someone might have alluded to what I'm going to ask, but I was looking at the draft guidance, and I was just wondering specifically why the current draft guidance is designed such that it's mostly for like standalone diagnostic purposes and doesn't necessarily include any screening or potential tumor genome sequencing. And I just wanted to get some clarification on why that was the case, if there's a possibility that maybe in the near future a draft guidance would be developed for that particular purpose.
Yeah, that's a good question and something we considered very carefully. I think the reasons were really twofold. One is, and I should qualify that this isn't really, if you read the intended use, it's not really for standalone diagnostic purposes. It's an aid in diagnosis. So, but that's something we also hope to get comments on during the public comment phase. But we chose this for two reasons. One is we believe that most of the uses are moderate risk, and that provides us with certain, that allows us to, we think we would rather develop this based, starting with lower risk, relatively lower risk uses of NGS, and then extend to the higher risk uses, of which we would consider, say, oncology, somatic variant testing, higher risk. The second reason is that technically we viewed this as probably one of the simplest cases. So again, we felt like this was sort of the core, recommendations that would probably be true for most tests. But then if we move to something like somatic, we have special issues of sensitivity, for instance, that we need to consider. And so we thought it would be valuable at this point to put this out and get comments on it while we are developing the specialized recommendations for, say, somatic testing or for risk or something like that. So it's not that we don't intend to do it and we would like to cover it. It was just a sort of practical decision in many ways.
Thank you. Once again, if you'd like to ask a question, you may press star followed by the number 1. Lee Lai, you may ask your question.
Thanks. Hi, this is Layli from Virtual Genetics. Thanks very much for giving me the opportunity to ask questions. I have 2 questions here. My first question is that I know the draft guidance specifically saying it covers whole exome sequencing and targeted panel. But I'm just wondering, how about whole genome sequencing, using whole genome sequencing to do the diagnosis? Was there anything that we expect a separate guidance, or there's something will be expanded?
Well, we intentionally separated out whole genome for now just because it's, it's slightly different in terms of its design. It's not that we don't want to encompass whole genome, but we wanted to, again, get comments on this more limited use before we start tackling any variation. And I think that was the reason. But we fully intend to expand to whole genome, even though it's probably not as widely practiced as targeted panels or whole exome for this now. It's something I think we could very well anticipate in the future. It's going to be more, as the costs drop and things get easier, it's going to be used more and more.
Thanks. And then I have a second question. So there is one of the slides that you've listed, different categories of these testing that will, in the future, we're looking for standards and guidance. One of it is a health lifestyle. Is that more toward direct-to-consumer service, or is that considered as a medical and similar requirement as the other disease area?
I think it was a little difficult to understand your question, but were you asking, if you were asking whether this extends to DTC testing, direct-to-consumer testing, or whether we intend to do that?
On the slide number 13, you have a separate block listing the scope of analytical stand. One of the blocks is health and lifestyle. So my question is that is the health and lifestyle considered as a medical condition?
Oh, lifestyle. I see. I'm sorry. It was difficult to understand you, but I think that yeah. So lifestyle, it's difficult and something we hope to get comment on. But I do think that you know, that it's certainly a use that is claimed for genetics and genomics. And I think, you know, right now, I mean, I would contend that there are some medical implication to that, even if the intent is to limit it simply to health and wellness or lifestyle. So that's something we want to consider more, and I think we will, you know, welcome comments on that. You know, we recognize there are going to be fuzzy borders around these bins, you know, and it can be difficult.
Thank you.
Thank you. And our last question comes from Nate Carrington. You may go ahead.
Yes, I have a quick question about the modification section of the guidance. So currently the modification section, it does not reference the K97 modifications guidance for deciding when to submit a 510K for a currently cleared device. It also doesn't reference the PMA supplemental process, so I was curious if not referencing those aspects was intentional and what your thoughts were on that.
Hi, this is Liz Mansfield. These two guidances were intended to lay out proposals for how we might look at analytical validity and how databases could be used as sources of valid scientific evidence. They are not intended to lay out all of the regulatory issues and requirements around making a submission for a particular product and updating that product over time. So did we leave it out intentionally? Yes, because we didn't really want to go into the discussion of every possible regulatory issue with tests, but it doesn't mean anything about the K97 guidance.
Thank you.
Thank you. And this time I'd like to turn the call back over to Irene Ihier.
Thank you. This is Irene Ihier, and we do appreciate your participation and thoughtful questions. Today's presentation and transcript will be made available on the CDRH Learn webpage at www.fda.gov/training/CDRH Learn by Thursday, August 4th. If you have additional questions about the draft guidance, please use the contact information provided at the end of the slide presentation. As always, we appreciate your feedback. Again, thank you for participating, and this concludes today's webinar.
Thank you for joining today's conference call. You may go ahead and disconnect at this time.
So we have